So the actual steps of DNA replication, as we mentioned, we start with the helicase and the primase together. You see they fit together. They're two enzymes, two proteins. They fit together, and they're going to work together. The job of helicase, as we said, is to unwind. The job of RNA primase is to put down these primers. And we'll look at where that comes into play and what that actually means for us. The other things we need to orient ourselves to are these Ssb proteins. These proteins are responsible for holding open the single strand. So they keep the hydrogen bonds from reforming, allowing the polymerase to come in and do its job. The polymerase, of course, adding the nucleotides. And then we're going to deal with some of these other words in just a minute. So DNA helicase comes in and unwinds. The Ssb proteins bind to those open strands to keep them open. So this prevents hydrogen bonding between the nucleotides. RNA primase is going to build primers. Notice it's RNA. Yes, we are replicating DNA right now, but we're going to use RNA. Why do you think that is? Well, it's single-stranded. This makes it very, very easy to work with. If we're going to use a template to build something and we already have one strand, having something single-stranded is much faster for us. DNA polymerase is going to add the nucleotides. When DNA replication is coming to an end, we can't leave pieces of RNA in here. So we're actually going to have to go in and cut out those RNA primers, and what's left behind are what we call Okazaki fragments. They are DNA pieces that are incomplete. Of course, discovered by a scientist named Okazaki. And DNA polymerase will come in and fill in the gaps. And then ligase comes in, and remember, ligase is the glue to make sure that everything sticks together for us and that we don't have any errors. So these Okazaki fragments, so in this case, these strands are RNA, and the strands that I'm circling are actually DNA. So if you come in and cut out the RNA, you have holes in your DNA strand. We can't have holes in your DNA strand, so we have to go back and fill in those gaps. So what's the purpose of these primers? The whole purpose of the primers is to provide the 3' end so that DNA polymerase can add the phosphate group attached to the sugar attached to the nucleotide, or the nitrogenous base, to be more specific. So, as we said, putting in these primers, the whole point of it is to give polymerase a place to sit down and do its job. Without it, remember this 5' end right here on the bottom strand, if we added another phosphate to the other side, that would be 2 phosphates side by side. We can't have that. So the whole idea here is these primers are built with a phosphate here and the OH at its end. So when polymerase comes in, it can add a phosphate to the OH as it should.